easier for me. All right, hi guys. Can you hear me up there? Good, Lily? Yep, perfect. Because I am not a loudspeaker, and I tend to migrate around, so I'm just gonna hang out here so you can hear me, because I think that's more important. So I'm going to talk to you about the research I did for my PhD, which was on quantifying labile organic carbon in sediments around Antarctica. I did this at North Carolina State University. I am no longer there. I am currently here. I am the lab manager and chemical hygiene officer, so I've seen a few of you guys around. But today I'm going to talk to you about my background and then this research, not about chemical safety. So I was basically born an oceanographer. Back in the 70s, oceanographer by the name of Robert Ballard made some huge discoveries about these deep sea hydrothermal vents and the biological community that were rampant there. This happened in 1979. It's the year I was born. Ballard went on to later discover the Titanic, but this is way cooler. A few years after that, there was a NOVA special that I saw. And from that moment on, I was hooked. I wanted to study the ocean. I did everything I could to be in the ocean. I read books, watched more TV, until I finally got to do my first research project, which was when I was in Bermuda, I was in high school, but this was at the Bermuda Institute of Oceanographic Sciences. That's the first research vessel I was ever on. It's the Weatherbird 2. It's now retired and just does kind of teaching. It's a teaching vessel down in Florida. I got to do some diving, and so here's some skipjack tuna that I took a picture of underneath a shelf. But I was really interested in marine biology. I got to study behavioral habits of octopus and squid in a variety of coral reefs around Bermuda. I wrote a paper. It was not published, but I was really excited. So I carried that into my first experience as an undergrad, and then I left school. After I got my associates, I needed money, so I went to work. I worn a lot of hats. I worked for a lumber yard. I was a head chef. I worked for Target Corporation. I did logistics at a computer storage facility. But all of the time, I felt that I was a scientist. Because to me, being a scientist is a philosophy. I could take things that I learned from each of these different jobs. No matter how disparate it was, I was using inquiry-based learning to inform my life and my roles in these positions. Till finally I had enough of improving processes for Target, and I went back to school. I started my undergrad at NC State in North Carolina, and in my junior year, I got an REU, so a research experience for undergrads. So is what Les was just talking about, like getting internships. This is the one that I had. I was doing geochemistry on sediments in the Horn of Africa, looking at plant leaf waxes and evaluating whether those could give any information about climate change in the Sahara. After that experience, I partnered with my future mentor. He had just come back from Antarctica on a huge leg of this project called Food Banks, which is a food for the benthic Antarctic continental shelf. This was an enormous multi-year project that I got to be a part of as an undergrad. And using that experience, I springboarded into what ended up being my graduate career. I went from marine biology into the chemistry. I was just absolutely fascinated by the idea that a sediment column could contain so much information about not just dirt. So this is what geochemistry in Antarctica looks like. You're taking cores, you're seeing all sorts of cool animals, you're working in dangerous situations, you get to work on massive vessels. But this is what geochemistry in Antarctica really is. Boiling dirt. That's what I did for 9 years. I wouldn't change it for anything. So these are some of the samples that I processed from the East Antarctic Peninsula. It's pretty boring, but it's also pretty awesome. So I'm pretty sure I'm the only person in this room that has been to Antarctica, I'm going to give you a little bit of background. So Antarctica, that's the south. Penguins, not polar bears. This picture, you can see one of 2 US research vessels. This is the Lawrence M. Gould. I did not do any operations on this. I was on the sister ship, the Nathaniel B. Palmer. So, penguins, not polar bears. It's very important. Polar bears are in the Arctic. So, why do I care about Antarctic sediments? Well, it ties into a global issue. We have climate change affecting the entire Earth, but not all areas are affected equally. This picture here shows the predicted air temperatures for the austral, so austral summer, or winters, I'm sorry, which is our summers. And you can see right here on the peninsula, we're talking about more than 12 degrees centigrade increase in air temperatures. That's 53 degrees Fahrenheit. So why is that important? Well, as the air heats up, that heat's got to go somewhere, and it typically will go into the oceans because water, being as amazing solvent as it is, also has an enormous capacity to hold heat. So the surface oceans are actually warming. That warming is reducing sea ice. That cascades. That has a change in the ecosystem. Those ecosystems are responsible for a lot of the chemical pathways that happen in the water column and ultimately I will investigate in the seabed. So we see these events in like mega scale proportions. So icebergs calving, things the size of cities. We have glaciers that are melting at unprecedented rates. And here is a photograph of the Larsen C ice shelf, which, this was taken in early 2017 where it was just fractured. And then by August of that year, it had completely split off. That's something the size of southern Wales. So these are huge pieces of the ice shelf that are disintegrating because our waters are warming. I don't care about that much. I care about what's going to happen to the carbon cycle because what I look at is kind of a net result. So from this figure, you can see that the anthropogenic CO2 has sources and sinks, but in the ocean is 25% of that sink. So if we start to change the interaction between the air and the sea, we're going to start to shift that sink. Perhaps more is going to get stored into the ocean. If so, what form is it going to be? Which brings me to my research on, I broadly call everything particles. So it can be organic or inorganic. So this is a floating sea ice mat. This is where a lot of productivity happens in Antarctic surface waters. So from the biological side, we have this high latitude food web dominated by diatoms and salps. All this biology ends up in this particle sinking. From the inorganic side, Most of the sediment is brought in by glacial till. So you have either alien-derived dust, surface particles, or things embedded that move out into the ocean. When that ice shelf, glacier, sea ice melts, you eject lithogenic material into the water. No? So as that material settles, it's a vertical transport, but we can also have sediment moving in from the other side, which is lateral transport. And just for clarification, when at your station you're evaluating if sediment is moving away, that's winnowing. As it moves towards it, that's focusing. So you have this particle, it rains down, gets to your station. Net accumulation of either lateral forces or vertical is the net flux, and that is what I measure. This is a sediment core taken from the bottom of the ocean in Antarctic on the continental shelf of the West Antarctic Peninsula. You can see this fairly compacted, homogeneous material. It's more of like clay, and this layer here is the result of a phytodetrital pulse. So that's all dead and reworked phyto and zooplankton that accumulate. The size of that is very variable and it's non-uniform around the peninsula. So we have to collect these cores. The ocean around the Antarctic Peninsula is fairly deep, usually 400 to 1,200 meters when you get to the slope. So we need to have very large vessels that can withstand both the impact of ice, but also can pull up this coring device from 600 meters down. So this is the picture of, that's actually me right there, guiding that Megacore. What this thing does is we lower it to the bottom of the ocean, and then these little tubes get pressed down into the sediment. When we pull it back up, there is a spring-loaded mechanism at the bottom that captures an intact core. This is a lot better than just dropping something free form, because that tends to disturb your sediment. And that layer that we see here and here is really important. There's a lot of geochemical data that we can gather just from those two barriers. So we use this device because it helps to preserve, and so we can get a near in situ sampling of the seabed. Processing it, though, is very messy. I am pretty much covered in mud all the time, or I'm covering other people in mud. Once I do that, you saw me boil in mud, did that for a lot, and then it's a little bit of chemistry, depending on what I'm looking at. So what do I actually measure? This is that little particle that I talked about earlier. Well, I care about radionuclides. So these are naturally occurring radioisotopes that either get incorporated into the tissue, as is the case with C14, or adsorbed onto the surface, which is the case with thorium, lead, and uranium. So each of these radioisotopes has a characteristic half-life. That half-life can be integrated over a certain amount of time to give information about what is going on in the seabed at different time scales. For this talk, mostly I'm going to be talking about the C14. So when we use C14 as geologists, we tend to look at sediment accumulation rates or mass fluxes. We want to know how has something been accumulating over time. And so that's what this profile shows you. You see there's error bars on this. and we don't even have the surface. That's because this tends to get smeared, and we lose some resolution in sampling. But like I said before, that interface there has a lot of information. Oh, here we go. This is the one I wanted. Here's a high resolution sampling that shows that surface interval, that upper 5 to 10 centimeters. You can see that curve. That curve is the basis for my entire dissertation. This one picture. What does that curve mean? Does it change? Is it static? Does it change based on the proportion of organic matter or inorganic matter? Like, this is where I talk about, like, asking questions about simple things can open up so many avenues. So this gradient here allows me to set up an endpoint with regards to the source of the C14, so that phytoplankton that's raining down, and the stuff that's at depth, things that have been there forever, reworked. And that is something that I'll evaluate in a mixing model. Why that works, to use the C14 of the plankton, right here you can see that this value is fairly similar to this body type tissue, so we know that whatever is falling down and reaching the seabed is being ingested by animals that live within the seabed. So this is my mixing model. I just define labile as something that's fresh that you want to eat, and as opposed to stuff that's old, reworked, and no one cares about. And I can use that to see how long it takes for carbon to work through that bioreactor. I think pictures are easier to understand. So that's something you want to eat. You want to eat the nice fresh pizza. You don't want to eat the leftovers. You can, but you're not likely to pick it. This is the stuff that's raining down, and this is the stuff that's been kicking around. So the other radioisotopes I use as tools to evaluate either that lateral relative to vertical transport, which is a measure of the inventory, so that total concentration of the radioisotope within that sediment column, or a measure of how quickly that sediment is being mixed. And so this is a holothurian, which is sea cucumber, sand dollar, and this is just a pictorial representation of what it looks like for animals feeding within a sediment column. So they're either scraping the surface and then burying a packet of food, or they're drawing it down through their tube, and sometimes that food is just raining down into an open cavity. But we can use lead-210 or thorium-234 as ways to quantify that mixing intensity. So in my research that I did, I had 3 objectives. One, I wanted to look at a newly collapsed and open system, which was defined as steady state or a non-steady state, and that was the Larsen A. Compare that to the steady state, which is along the West Antarctic Peninsula. And then I wanted to find a way to, like, ground truth, to check whether or not my reasoning in how I'm setting up this definition of lability would hold against another method. So, non-steady state, that's a nasty equation. I don't like it. It's not fun. Steady state, the only difference is, does the concentration change with time or not change with time? So, no, not good. Pictures are still better. So steady state, you're always getting your food. pizza's being delivered. Non-steady state, you're just sitting there waiting, you don't know. You could starve, you could not. Much easier. So this is just a Google Earth view of Antarctica. This side is the non-steady state side. This side is the steady state side. So that's the Larsen A ice shelf. It's collapsed. And this is the study area for the West Antarctic Peninsula. A variety of different environments were sampled on the West Antarctic Peninsula just to get a sense of, does your depositional environment affect lability? And then on the non-steady state side, I did a kind of a time series analysis. And what that means is these stations that you see here were designed to represent where the ice shelf was at a given period of time. So this is a USGS map that shows the ice shelf being here at 1945, 1968, and this is 2000. So this ice shelf collapsed catastrophically in 1995 and completely in 2002, but it had been eroding for the last 150 years. So that non-steady state system, we wanted to look at the labile carbon distributions. Does it vary with time? Does it vary with space? And to do this, I had to use lead-210 in that non-steady state solution. So again, mixing model. I'm evaluating the food you want to eat relative to the food that's just been passed over many, many times. So what do I mean by the non-steady state in terms of this system? If my study site is here, I am not getting any biological material raining down, therefore my C14 content is just going to be old and reworked. But when that ice shelf collapses, now we have productivity in the surface ocean, we're uptaking CO2, making biomass, and that's raining down. So we now start this cycle. So we should be able to see the pulse of C14 reflecting this change in regime from this oligotrophic biological desert to this bio-rich evolving ecosystem. And that looks like in a sediment column, you have this old stuff when the ice shelf is present. As that ice shelf is broken off and your surface waters are becoming productive, you have this ingrowth of this labile fraction, and eventually it starts to make leftover pizza down here when before it was like really, really old. So quasi-time series, my station started from 170 years. I went down to 15 years post-collapse. And this is a characteristic C14 profile for the Larsen A. You can see that shape that I talked about, where you've got decay or mixing within the upper 15 centimeters. So that gradient helps to inform reaction kinetics and potentially use that to separate complex carbon mixtures. So in the past, C14 was not used, partly because of analytical reasons, but partly because we had a measure, we have several measures for lability. Some people will use total organic carbon to evaluate whether or not carbon is being used and converted within your sediment and biological reactor. But there are some complicating factors. You can't date the C14 of, total organic carbon. You can't guarantee its source, so you can't evaluate whether you have lateral transport. But with the C14 model, knowing that your C14 signature can tell you about source, and the depth of penetration gives a sense for how much of that sediment layer is being reworked, it becomes a much better way of evaluating the amount of labile carbon in your sediments. So this is lead-210 inventory, and I told you could use that as a way of evaluating lateral flux. You can also use it just as a bulk indicator of sediment transport. So you can see that there is a lot of lead-210 accumulating further off shelf where you've had longer communication between the surface oceans and the seabed, and less as you get closer to the continent, where the seafloor has only been communicating with the surface ocean for the last 15 years. LOC inventories, similar trend. Your most buildup of labile carbon happens outside where the ocean is kind of modulated now by seasonal ice rather than the ice shelf. And that decrease as you move along indicates that, yeah, the LOC requires time to build up within the seabed. But the fact that it is building up is important. It means that the biological community in and on the seafloor is not adapted enough to be able to process all of the carbon that's raining down. So you're creating another bank of food. Because that labile carbon is building up in the seabed, it means it's going to last longer, and it takes longer for it to react and pass through that biological reactor. So the further offshore you are, you're actually seeing that it takes longer for the carbon to pass through that and is becoming older. So this is where your leftover pizza is more likely to be. And again, this newer communication with the seabed and the open ocean, you've got smaller inventories, but it's being processed a lot faster. So what I just said in summary was bulleted right here. You've got LOC accumulation in all stations, but as you get further away from that ice edge, you get a buildup of older. It's still labile by my definition, but it is less likely to be picked up. But we also see that transformation from that older reworked carbon to a more sustainable seabed. Then we move across the peninsula to the west side, and here I was looking at the same parameters, but in a sense trying to evaluate, do depositional environments affect C14 and LOC? So west side sampled fjords. This is the Palmer Deep. It's a 1,100 meter depression, an embayment down in Marguerite, and then a few transects across shelf. So here are all of the C14 profiles that I generated for these stations. These features in the upper 10 centimeters, pretty much what I showed you in that very first slide. There is a lot of reaction kinetics happening. Once you get below that, you start to see a change, and this is where we start to move from the very labile to the semi-labile. So remember in that figure where we started to build up more and more labile carbon, and then that semi-labile fraction ended up becoming the bulk. That is what we're noticing here. Everything below this upper 10 centimeters is reworked, but it's still more labile than the sediment that's been kicking around for thousands of years. So this is just turned sideways. North is to your right. There's really no spatial pattern in LOC inventory when evaluating all of the stations together, but we do notice that the deeper you are, the larger your inventory. So that Palmer Deep acts as a focusing center. So we see an accumulation in carbon inventory. We also see that in the lead-210 inventory. We see that as you move across the shelf, you tend to increase in inventory, and that's again lateral transport. The coastal waters are more bioactive. Sediment gets reworked and moves onshore to offshore. So it will tend to move in this direction, which makes sense when you look at the differences between some of these inventories. For the West Antarctic Peninsula, I was able to use 2 different radioisotopes to evaluate mixing. Thorium-234 has a really short half-life, so you can look at what is going on in terms of months to a season or a 100-day time scale. And then the lead-210 will allow you to look at longer term mixing, so tens to hundreds of years, or well, up to 120 years. So again, no spatial pattern regarding the turnover time for lead-210 or for LOC on seasonal timescales, but we do notice that every single point is at or greater than 1. In terms of years, that means that if you live in the seabed, you don't have to starve when productivity goes dark. So you can feed year-round on labile carbon based on our data, which corroborates my mentor's earlier study for the food banks, where they had done their own evaluation, not using the C14 model, but came up with very similar numbers regarding 100-day timescale lability. When you look at, as the carbon moves through that reactor and you get below the depth of penetration for the thorium-234, we evaluate the labile turnover times on 100-year timescales using lead-210. And what you notice immediately is that these are orders of magnitude greater than with the thorium-234. Part of that is tracer dependent, so 23-year half-life, things are just going to be mixed much more slowly. But the other part of that is it's actually showing you what the particles within that sediment reactor are doing, which is being mixed slowly. They're already reworked. No one wants to go back down in there to ingest it. So that's what the other half of that story. The other reason why you see carbon that goes to the Palmer Deep is going to last 4,000 years. So in summary, they really not a spatial relationship on mass between the different stations and LOC characteristics, but we do tend to see that similar aging effect. When you last longer, you're in a deeper environment, further offshore, we get a buildup of the semi-labile on the West Antarctic Peninsula, something that has not really developed on the East Antarctic Peninsula. Check the time. So the last part of this talk has to do with a method comparison. So it's all well and good to just come up with a model. Those were really arbitrary, saying that the labile is my phytoplankton, the refractory is whatever is at the bottom of my core. My cores didn't go deep enough, or maybe somebody's eating something else. So you have to test your hypotheses. And so I did it via not replication, but ulterior method. So I did ramp pyrolysis oxidation, which was really nice because I didn't have to do any of the chemistry. I just processed data. But in essence, they're taking my samples, they incinerate them, then they send them through a chromatography column. Based on fractions, they evolve the CO2, and I get a nice temperature CO2 plot. So I only had funding to do one station, so I chose Station G, which was that East Antarctic Peninsula had only been communicating with the surface ocean for 15 years. But the reason why I did it is because there was the biggest separation between the surface C14 value and that at depth. So my surface value, the bulk C14 signature, was about 5,050 years. At depth, we were right around 30,000 years. So almost to the limit of detection for C14. So pretty good separation. If I was going to find validation for my model, I figured this was going to be my best shot. And here is the evolved CO2 temperature thermographs that I got. So surface, mid-depth, and bottom of my core. Just to show you your way around a thermograph, the lower the temperature that you see a value, the more labile, more reactive, smaller it is. And conversely, when you get higher temperatures, it means it is less labile, more unreactive, larger molecules. So we can see two nice features, one at a lower temperature, one at a higher temperature, and the relative proportion and temperature at which those two peaks came off at evolved down core. Awesome. That's a success in my mind. It looks like we've got a labile peak and a refractory peak. I worked with a researcher down in Florida who shared his MATLAB model, and we worked on deconstructing those C14 peaks into there we go, into these multiple components. And so while it looks like there's two components, the best mathematical solution actually comes up with three components. And so I was able to look at not just the C14 content of each of these fractions and where they came off of, but actually put some other isotopic data in there and come up with these components through the MATLAB analysis. And this C1 peak, that is my most labile fraction. And the C2, that's my semi-labile, and that C3 is my refractory. When you look as you move down core, the total amount of the C1 peak increases, and it actually moves a little bit more higher temperature. So there is a conversion of this very fresh material into more refractory. And we start to increase the amount of semi-labile and ultimately refractory. So at the bottom of my core, there's practically no labile carbon left, and we have a huge amount of refractory carbon. Awesome. Another like plus in my models column. So the last thing I did for this model comparison was look at how does that C1 fraction really compare when we look at it on a absolute scale. And so my bulk C14 signature is here in blue, my ramp pyrolysis is in red, and while I don't have as many data points for the RPO fraction, like there's pretty good agreement. Like you've got the general shape is still there. You may ask, well, why does it go to 0 at depth and it does not go to 0 here? That's just how my model defines labile carbon. At some point, I have to have none. So yay, my model works. That was like really exciting. It was validation that at least in that extreme situation, all of the work I'd been doing for the last several years was paying off. So overall, why I care about this is because high latitude environments are changing rapidly. And while that's important on itself, we don't understand these environments to begin with, and now they're changing in ways that we can't really quantify. So as much information as we can gather about these kind of trends in carbon cycling is important. There's actually not very many studies that deal with carbon in sediments. Most people deal with water column. So if only it were fancier and flashier and I wasn't just boiling dirt. And so I use my radioisotopes to evaluate these processes on different timescales. And hopefully I'll be able to continue that in the future. And so I could not have done this without my friend and mentor, Dr. Dave DeMaster, or Craig Smith, or Enrique Isla. The sampling wouldn't have happened without the Nathaniel B. Palmer or their crew. And I couldn't have done any of the RPO stuff without the Woods Hole Oceanographic Institute, especially Mary Lardy, who actually processed all my samples, and Brad Rosenheim down at Florida State University for his MATLAB code. I'd like to thank Les Kanat for inviting me here and all of you guys for listening. So with that, let's take any questions.
Hi, Lily Dunbar. I'm wondering, is this carbon that's going into the ocean and getting turned around and everything, is that coming from the atmosphere mostly?
In Antarctica, not directly. So if you look at the C14 age, like the reservoir age around the Antarctic Peninsula, it's not reflective of the C14 reservoir age of the atmosphere. Like our current atmosphere has a, because of atmospheric testing of nuclear devices, has a greater than modern C14 value. The reservoir age in the Antarctic is anywhere from 800 to 1500 years. So if you, like, I didn't point it out in my figures, but my plankton value is 1300 years old, but it's brand new. So not in the Antarctic, but in other environments, yes.
Why on the stable side your points were very spread out, but on the unsteady side they were all very concentrated? Why wasn't it more spread out on the other side?
For two reasons. So in the non-steady state side, what I didn't put up there is a map of what's called a long-term ecological research station, or for the West Antarctic Peninsula. They have this 100-kilometer grid network that my team was sampling over top of. And so they were kind of predetermined for us. on the West Antarctic Peninsula, or I'm sorry, on the East Antarctic Peninsula, part of that was by design and part of it was by happenstance. So the original cruise was supposed to go to the West Antarctic, or further south to the Larsen B, got frozen out, couldn't get to it, so they decided to do the Larsen A. And then they had to just make up sampling schema on the spot. So they decided to just do a transect, and then they made the designations for each of those stations based on that sea ice, or sorry, the ice shelf edge map. So you go to sea with the best of intentions, and sometimes you don't even get to do science.
Hello, my name is Ashton. How long is the time process between digging the mud up and setting it up the lab?
Depends on what you're doing. For thorium-234, because it's got such a short half-life, it's actually got to be processed really quickly. So for that study, I collected them and processed them all within 30 days. But I could, for lead-210, long half-life, I could wait 30 years before. Most of my stuff was between 8 years and 7 days old. Yes?
Where did you get the equation? Where did you get the equation for the steady versus unsteady?
For the what?
The steady versus unsteady?
The non-steady state differential equation?
Yeah.
It was given to me. So it was worked out on the back of a napkin using the diagenetic equation for the mixing of a non-conservative tracer over a series of time. And one of the pieces in there was this Taylor expansion. I can't do that math, but he could, and so I just trusted Jerry to steer me right. Yes?
My name's Alex. What was your sample size like? Because obviously you're limited in how often you can get out there and grab stuff and how much you can take with you. So.
Yeah, a lot of times sedimentary geochemists have a sample size of 1. I would try to take as many replicates as I could. Now you saw that rosette that we sent down had 12 megacores on there. I've got to share those with people. Typically I could get 2 cores from that cruise. Other cruises that were more radiochemistry oriented, I've gotten as many as 6.
Were they fairly consistent in looking at temperature versus everything that you from each different location, just based on looking at it, or was there a lot of variation?
It's fairly uniform, but there is a lot of variability depending on depositional environment. The shallower you are, the sandier you are. We tried to sample deeper environments that would just be collecting clay, and that has to do a lot with the fact that clay likes to adsorb things onto its matrix and collects radioisotopes much better.
Just Patrick Austin, generally what were the days like when you went off and researching, from when you wake up just to when you're back?
Let's see. Well, given that I was there in the summer, we've got 30 minutes of twilight, so there's never a night. Shipboard operations are already chaotic, so there's no real schedule. Weather-wise, though, it was quite pleasant. Think Cape Cod in winter, so the air temperature was just about freezing. It would snow most days. When it wasn't snowing, it was just gray.
I'm Melissa. Was there anything outside of the research that you found pretty interesting in pulling up from different samples that didn't pertain to what you're studying, but was just kind of like interesting?
Yes. I don't think I have a picture of it. We took one core from the continental slope. It's not something you often do with a megacore, just because the continental slope is like this, your megacore comes down and it wants to just tip over. But we were able to do one cast, we just did it on a lark and it was successful. Pulled it up, it was this like nasty, sandy, rocky core, but it had a animal in there that none of us had seen before, and in fact had only been documented one other time. It was some sort of like naked mollusk thing, technical term. Yes?
Les Kanat. So, I know why I like hot, crispy pizza. But what is it about the organisms that prefer the labile carbon over other carbons, and what is it, labile carbon, really?
Labile? What is it?
Yeah, like, wow, what is it, why is an organism, how does it differ from old pizza? And like, why would an organism prefer that type of carbon? Because, pizza's pizza.
Right? You would think pizza's pizza. So one of the reasons why an organism might prefer the labile to the semi-labile may be from a thermodynamic standpoint. So the more reactive something is, generally the easier bonds are to cleave. They can strip off hydrogens really easy. They've got functional groups that are awesome. The other reason could be they just have adaptations for it. I can quickly get this, and therefore I'm going to take it away before someone else can. In terms of my research, I do not know exactly why. It's something that is an ongoing question. Like, what exactly does it mean to be labile? And mine is an operational definition. Like, I'm saying that we see C14 of plankton in the gut tissue. We're going to call that the most reactive. It doesn't have to be. It could be something else. And labile even differs in different environments. So it's a continuum of reactivities. Like, you can actually start outside our doors here with carbon that is reactive, and it'll make its way eventually to the ocean. Does it mean that whatever is outside our doors is more reactive and what it goes to the ocean is unreactive? No. We have like changing community structure, we've got changing aqueous environments. Like, there's so many things that play into the idea of lability. You just either have to confine it to space in either a horizontal or vertical sense, or confine it in time. Any other questions? Kara?
Can we go back to the picture of you with the ponytail?
Yeah? What is that? That's going to take. We're going to have to do the talk again then, right? You just wanted to see her mud on her face? Is that it?
No, I'm questioning your choice in hairstyle.
It's non-functional when you're trying to do high precision chemistry, hence the ponytail. It's actually in a bun.
I'm surprised that your
the woman working with you doesn't have her hair in a ponytail, to be honest.
She did.
She did.
She was not actually working at that point. Yes. Well, I was working, and she thought that she could, like, hit me in the face with mud, but that didn't go very well for her. I got a student. Sorry, Les.
Hi, my name's Elizabeth, and I was wondering how many places have you been during your research collecting the samples?
Collecting? Does that include non-exotic stuff? Sure. Hundreds.
What has been your favorite place to collect from?
Probably Bermuda. That's pretty hard to beat. Yeah, probably if I got to do more off-ship operations for the Antarctic, it might have been able to beat it out, but I always stayed on the ship, so. Yeah, Lily?
So you have a background in marine science, and you've done all this amazing research, you have your Ph.D. What are you doing here?
Huh? What am I, like, this is what I want to do. I want to share with undergrads. I want to be able to inform, like, small-scale science. I started, like, my college career at a community college, and I got to do, as part of a work study, the exact job that I'm doing now. I was running a lab, like, I was prepping things, I was, like, helping students, even though, like, I was still a student. Like, that to me is part of science, is that communication, that handing off of knowledge. That's why I'm here. Plus, you can't beat Vermont winters.
So this, these data you collected are related to the most recent ice age? Can you say that?
I refuse to say anything about my data relative to the Holocene.
Why?
Because I view C14 data more as an information regarding dynamics than I do as age dating. I think there's a lot more quote-unquote truth to be gained from looking at it as a process than as a tool. But yes, you could say that this is Holocene era.
So in previous ice ages, are there similar results?
Would not be able to tell because no way to evaluate mixing. You had your hand up.
Yeah, do you get seasick at all?
Yes. The time when we crossed the Drake Passage, so you can go like this on a wave, you can go like this on a wave, or you can do like this on a wave. We didn't realize that we had made it out into the Southern Ocean, and we were up on the 4th floor of the Palmer. So when you're up there, like one movement, little movement is really exaggerated. So that was awful. That was like 30 some odd hours of puking.
So that made the mud look good.
Oh yeah. Luckily we hadn't started sampling yet, so.
All right, well, thank you very much.
Thank you guys for good questions.